Welcome back to MACNA 2020. I'm your host, Kevin Erickson. Today I have the honor of introducing Dr. Andrew Rhyne. Andy has invested his career in understanding marine ornamental aquaria, the biology of aquarium fishes and invertebrates, as well as the industry driven by human fascination for these creatures. The trade in aquarium species can be considered a data-limited industry, and the lack of available trade data hinders sustainability movements within the trade. Andy has worked to develop solutions to these data gaps. He has worked to advance the field in marine ornamental aquaculture, developing methods for breeding and rearing popular species in aquaria. Andy's body of work illuminates life histories, husbandry and larval rearing requirements of aquarium fish and invertebrates. He has pioneered methods for culturing calanoid copepods and has greatly increased the success of hobbyists and researchers with small tropical marine fish larvae. Here to talk about Caught in the Act, Fraudulent Data and Activism, Slow Progress in Cyanide Detection is Dr. Andrew Rhyne. Andy, welcome to MACNA 2020 Phoenix Rising.
Well, thank you so much, Kevin. Thanks for the great job hosting and the video editing.
You're welcome.
All right. Well, today I'm going to talk to you about some work that we have been doing for the past 5-6 years or so, and I've given a couple talks at previous MACNAs on some of the data behind this. And today I want to kind of give a sum of where we're at and show people kind of where we've come and what we kind of know now, and put the pieces together on some of the other information that is out there, and try to provide some evidence for a little bit of concern about a few papers, and also some direction as to where we might be going. With all science and projects, our work is a large collaborative effort. And so I just want to acknowledge my co-authors, Dr. Nancy Breen, who's a spectacular analytical chemist, who's been working with me for the past, since about 2014 on this project or so. And we've had a couple, a graduate student who was an undergrad in my lab, and then went to University of Massachusetts at Boston with Michael Talussi and did his master's on this project, Alexander Bonanno. We've had a ton of undergrads work with us on this, and we've got an exciting collaboration with Mystic Aquarium. Most importantly, I want to acknowledge some of our partners. Dominion Diagnostics is a laboratory diagnostics company here in Rhode Island, and they have been really great in supporting us with their next generation ultra HPLC technology. Importantly, I want to acknowledge the funding source that I've received for much of the work you're going to see today, and that is through the Pet Industry Joint Advisory Council. PJAC has been very interested in supporting this issue and trying to resolve some of the problems around cyanide detection to increase the sustainability of the industry. And so they've been gracious enough to support that. And again, Dominion Diagnostics, who has provided thousands and thousands of dollars worth of instrument time to us to be able to tease out what's actually happening in fish. Many people are aware of the problems with cyanide collection. They've heard about it, or they've seen news articles about it. The most high-profile article that has been out in the last 5 or 6 years was an article that ran in 2016 in National Geographic titled "The Horrific Way Fish Are Caught for Your Aquarium with Cyanide." And what really struck people in the industry was the byline, or this subheading, where up to 90% of the saltwater aquarium fish imported in the United States are caught with cyanide. And then you'll notice it says a new petition is calling for a government crackdown. And this really raised people's eyebrows a lot, because 90% is a massively high percentage of number, right? So it's a large percentage. And it would basically mean that almost all the fish that you buy were caught with cyanide. And this really caused a lot of problems and consternation out there. My phone was ringing off the hook for several weeks when this article came out. People really wanted to know what was going on. And it turns out that that's not a factual statement. That is a misinterpretation of a government report, but it did a very good job for the people who had this article pitched in National Geographic. And that was to stir up some controversy and raise some headlines and PR for their effort. So that story highlights a study that came out that really was an exciting study when it was first published, and that was this idea that you could take the water that fish were being held in, and you could sample that water and you could tell if they had been exposed to cyanide by looking for a metabolite called thiocyanate. And there's a lot of chemistry and biochemistry involved here, but essentially any organism that is exposed to cyanide has to detoxify the cyanide, and that product that is left over, or that metabolic byproduct, is called thiocyanate. And all animals do this because all animals are exposed to cyanide at some level from the environment. So cyanide exposure is a natural phenomenon that happens in the environment, but it also is an unnatural phenomenon. And the best example I can give to people is that if you smoke cigarettes, right, burning products will produce some level of cyanide. Firemen are really susceptible to cyanide poisoning. So if you're inhaling cigarette smoke, then you're inhaling a certain amount of cyanide, and your body has to deal with that, and it converts it to thiocyanate with the same processes and biochemical processes as fish. And so the same metabolite is in your saliva and in your urine, thiocyanate, so you can actually measure that. When this paper first came out, it was really, I mean, it was very exciting. Rhett Talbot wrote this article in Coral magazine about it. He had contacted me and asked me to comment on it. I had known about this paper since about 2009. Ricardo Collado and I have been long colleagues and friends, and we had worked together in graduate school, and we were at a meeting in Veracruz, Mexico. Oddly enough, it was also postponed because of a pandemic, the swine flu. And we were sitting down. I'd just flown in, and we were talking about our labs and what we were doing. And he was telling me about this chemical test that some colleagues of his had developed in the chemistry department at his university, and he was really excited about it. And so when the paper came out, I was like, you know, I've always been one of Ricardo's biggest fans, and this paper was it was stunning, right? And the best way I could describe the study was it was just so simple and elegant. Right, and I was certain you would see a ton about this, because knowing the lab it was coming out of, how prolific they were in publishing papers and doing research, I expected to see a lot of papers working on this. Just to give you a quick background on the process, essentially, if you expose a fish, and they use Clarkey clownfish in their study, if you expose a fish to cyanide, that fish will take that cyanide in either through their mouth as they're drinking seawater, so saltwater fish actually drink salt water, or through their gills, right, as they're bringing in oxygen. And then there's a process where that fish will have to detoxify that toxin, just like you would if you smoked, and they turn that into thiocyanate. And so the paper's premise was that if you held these fish in water, and you change that water every day, then you would be able to detect that thiocyanate coming back out of the fish if they had been exposed to cyanide. Really powerful idea that you could, in essence, you could detect cyanide exposure by holding a fish in water and taking a sample of that water and running it through an instrument and looking for thiocyanate. This meant you could hold the fish for a day, sample the water, and then the fish could go on its way, on its journey, and it was a non-lethal method for detecting cyanide exposure, which was really exciting. And so I just want to go over a few important points of the work. And mostly, I want you to be aware of a couple really important details. And one of those is that they looked for 28 days. So they had those fish in the glass jar for 28 days. They changed they basically did a 100% water change, or moved the fish to a new jar. Not exactly which one they did, but they changed the water out completely every day. And so they renewed that water. And so every one of these values you see are the amount of thiocyanate that they were finding in the water after 24 hours. Right? And you can see that they had two exposures: a high cyanide exposure of 25 parts per million. Don't worry about those numbers, just remember 25, right? And a low or medium exposure of 12.5, so half of the other concentration. And this is very common in experimental work, where you do a treatment and do half of that treatment. But importantly, they had a control. And that control was a fish that were not exposed to cyanide. And that's very important also. So they go through the experiment. They sample the water every day. They measure the water, and they find thiocyanate coming out of the fish after 2 days or 6 days, depending on the exposure. And it just keeps going up, right? And it increases and increases and increases until it plateaus at 21 days after exposure. And their last day of sampling was 28 days after exposure. This is a long time. And this is really important because at 28 days post-exposure, these fish would be in the source country after collection, right? We know that. We know it can be a long supply chain, but we know that it takes a week or 2 weeks for fish to be collected and moved into the country they're going to be imported into. And so theoretically, based on this paper, you could actually check fish in the country where they were coming, right? And it looks very stable. And we'll come back to this. We'll come back to this figure in a little bit. But this really made people in the industry really nervous. And you can understand why, because they don't have control of their supply chain necessarily. And there's always a concern that someone is fishing with cyanide in some countries where these are coming from. Different importers do a different job in trying to reduce that risk of cyanide collection in their supply chain. Some importers are very bad about controlling their supply chain, and some do a great job, right? And I'm happy to chat with people about that. But there's an unknown factor, right? Where these fish are coming from 9,000 miles away or so. So you really aren't on the boat when they're collected necessarily. So I was headed to this International Coral Reef meeting, really excited about this talk that I was going to see. We have been working on this for about 4 years or so, and it's always good to see other people working in the same area in science. And so we have been struggling with this method, trying to reproduce the data, and so we were obviously very interested. So I'm sure some of you have recognized the names of at least one of these individuals in this presentation. That's Renee Umberger of For the Fishes. And so she's a well-known anti-aquarium trade activist who's worked for many years to put pressure on the Hawaiian fishery and has successfully really curtailed that fishery and caused some of its closures. And then Craig Downs is a environmental toxicologist who has done quite a bit of work on sunscreens. You're familiar with his work if you've seen any of the bans on sunscreens because of the fear that they're going to kill corals and some other things. So, these two individuals gave this talk, and I'll get into that in a little bit. So in this talk, they made some really extraordinary claims. We were just blown away by the claims that they were making in the talks. It was extraordinary. Right? Craig Downs gave a very convincing explanation of their chemistry, and they showed that they had 51% of the fish that they tested positive for thiocyanate. And so based on that, they're saying that 51% of the fish that they tested were caught with cyanide. And those range from a low prevalence of 29%, right, to a high prevalence of 81%, which was, again, just absolutely shocking. And the fish that probably drew the most attention to us was the green chromas, right? So they're claiming that 81% of green chromas were collected with cyanide. And I got on Messenger at the time and started messaging friends in Indonesia during the middle of the talk and asking them if they knew anything. And I got some messages back that were like, it was just shock. It was just really, really, we were quite shocked. So, importantly, after or about the time of this presentation, and I can't remember if it was, I think it was just before the talk, the conference, right? They released a press release that basically said that 50% of the saltwater aquarium fish in the United States were exposed to cyanide. And again, a lot of consternation in the industry about this, right? Everybody's known cyanide issue. Cyanide has been an issue from certain countries. We just don't know the level of issue, and different importers do a different job trying to curtail or reduce that risk. They included in their original press release some data. And so this is just a chromatogram to show that they could actually do this and repeat the method, basically, to give you some idea that they are making a valid claim. And importantly, they included a table with all of their concentration data. And I'm going to come back to this data in a little bit, but this is actually quite an important piece to the puzzle. So Umberger and the Center for Biodiversity, so Center for Biodiversity and For the Fishes, along with some other collaborators, petitioned the United States government. They produced this report called Poison Waters when they're talking about how cyanide fishing and the aquarium trade are devastating coral reefs, right? Again, raised a lot of attention, caused a lot of concern in the industry. At the same time, they petitioned the United States government to essentially require National Marine Fisheries Service and the U.S. Customs Office to test fish and implement some type of cyanide-free certification scheme, right? And of course, the method they told the government that needed to be used was this really elegant method where you could hold the fish and sample the water. And so that presentation at the Coral Reef meeting and the white paper Poison Waters and this press release all kind of come together to put pressure on the United States government to look at this issue and potentially reduce the imports of aquarium fish coming to the United States. Many people listening to this talk will be familiar with these groups. The Center for Biodiversity has worked for many years to list species on the Endangered Species Act. Most aquarists are familiar with the Endangered Species Act because of the proposed listing of 82 corals back in around 2009 or so. And that process, ended up causing the listing of a large number of corals in the hobby as threatened. And there was a lot of questions about the data behind some of those assessments. So that was really the goal, I think, of their work, the outcome was to petition the United States government. And again, you can imagine the response from 104th Street on this with the importers in the United States. And Rhett actually interviews several of them in this article. So, again, we've been very interested in this topic. Again, I knew when this paper came out, other people would be interested in it. And that was just a given because it was such an important paper, right? So one of the most important things that happens in science is the replication of others' work, right? And I don't necessarily mean the duplication, doing exactly what other people do, but I mean replicating other people's work. So you take a fundamental idea that somebody has proposed and you repeat an experiment to demonstrate that their idea is actually possible, or maybe it's not, or maybe there's some other subtext to that idea, and you kind of build on everybody's work. And that's why you're able to see these amazing leaps in technology that we're all experiencing today, whether it's being here on Zoom or your iPhone or your aquarium, because things have happened over time that have built up, and this knowledge base has built up over time. And that's because people are repeating people's work. The best analogy I can give to people how this works and how hard this can be sometimes is, I don't know about you, but I bake a lot of bread now because, it's a pandemic and it's one of those things you do. You can take a recipe for bread and you can make fabulous sourdough bread, or you can make really terrible sourdough bread, right? So you could be a really great baker, give someone in their house the same recipe and they do a really bad job with it. So there's often a lot of work that goes into replicating somebody's process to get it to be the same or similar, but it can be done if the experiment is really well laid out and the methods are really well known, right? And so we set off to do that. Again, we were really excited when this paper came out. I knew it was coming out, and I had found some funding to help us buy the equipment we needed. I roped my friend Nancy Breen and the analytical chemist into this, and we got some students, and off we went replicating this. We were not alone, right? Again, when important papers are published in science, other people are interested to validate the work, right? And I'll introduce Sebastian Freeze to you. Dr. Sebastian Freeze is a marine ecologist, marine biologist out of Germany, very interested in Indonesia, has done a lot of work on coastal resource management in Indonesia and with fishermen, got a really long body of work in that area. He was the editor of this paper that was published in PLOS One, excretion of excreted thiocyanate detects live reef fish illegally collected using cyanide. So he was the editor of this paper. His job is to handle the review process, find reviewers, take those reviews, talk to the author about those reviews, and then get that paper kind of peer reviewed and out into press or reject the paper. And so Sebastian was very well aware of the work, obviously, and when it was published, he dispatched a master's student to Indonesia to collect water samples from fish where they knew cyanide fishing was happening, and then shipped those water samples back to Europe to be analyzed. Now, Sebastian was also at this really, really important meeting, this in Coral Reef meeting. And so we are sitting in the audience together. We've known each other for quite some time. We see each other at meetings throughout the last 20 years or so, or 15 years. And, we were very interested in the data that was being presented because we were both working on this topic and we were both having problems. And so after the meeting, and one reason meetings are so important is because you can talk to people who are working in similar areas, and you can advance your own research by getting some insight of some of those small little details, about how much flour do you put in the bread, or how long do you mix it or knead it. Those things that maybe weren't in the recipe, but you need to know to make really great bread. And their answer to us was really quite striking. They had no good answers as to why they were able to do this and no one else was. And they basically said, you must be doing something wrong. And that raises, your eyebrows a little bit when someone tells you they can do something really well. It's very easy, but they can't tell you how to make it work. And so Sebastian published a paper, and his group published a paper, and their conclusion was, it doesn't work. Something's wrong, right? That's basically the conclusions. You can read about this. South Pacific Council's Live Reef Fish Information Bulletin has been around for a long time. Very, very important publication series. It's considered to be gray literature, but I consider it to be some of the most important publications that have been published about the South Pacific, right? It's very tailored to their group. A lot of authors are from those islands, which is really important, and they talk about local issues. And so he published this. Natural to publish this there. They didn't have a lot of data. It turns out they had a lot of trouble getting the data set from their collaborators that they were collaborating with in Portugal, who did not want to release the data. Sebastian, I'm sure, would talk about this at some point. But very frustrated, really frustrated senior author on this paper. She was a master's student that basically, and Rhett talks about this in an article I'll show you in a minute. This basically burned her out of science trying to replicate this work. So we saw Sebastian's paper come out. Nancy starts working on the math on this because we're really concerned now about this data. And we publish a paper after 4 years of work. A lot of work went in here. And our conclusion is that their test, their proposed method, is not plausible. It's just not possible, right? And I'm going to go through that really quickly with you. I'll warn you, there's a little math here, but not a lot. And then I'm going to show you some really interesting things about their data. So here we have our fish, right? We have our little Clarky clownfish, right? Our little Clarky clownfish is in a large amount of water, 1.5 liters of water for a 2-gram fish. And a 2-gram fish, just to give you an idea, is a fish that's about an inch or so. Really small fish, right? And this is a figure 2 from the paper, right? And you'll see the bar graphs again. I introduced this to you earlier. So we took I basically reconstructed the data. I took that bar graph and I calculated how much thiocyanate the fish had to have excreted to generate that bar graph. And that comes out to about 240 micrograms of thiocyanate. Now, you don't need to care about what a microgram is, but just remember that 240 number, right? Okay, so now that's thiocyanate, right? And we know that's the metabolic byproduct of cyanide exposure. All animals produce thiocyanate if you expose them to cyanide. That's one of the pathways of detoxification. If you convert thiocyanate to cyanide, right, and this is a very generous conversion, you get 107 micrograms of cyanide that fish were exposed to, right? Again, you don't need to understand what a microgram is. It is a very small amount, right? But 107 is about how much cyanide they would have to have been exposed to generate 240 of the thiocyanate. Right? This is just a simple chemistry formula that all freshman chemistry majors basically do. So the questions you have to ask, knowing this number, is can a 2-gram fish take in 107 micrograms of cyanide? Is that possible? Right? And if it is possible, does it really, what is that going to do to the fish? And so you have to ask the question, is 107 micrograms of cyanide a lot? Right? And that depends. It depends on your body mass. If you're an elephant, 107 micrograms is really not that much. But if you're a tiny little fish or a little small mouse, 107 micrograms might be a ton, right? And it turns out that it's not possible for a 2-gram fish to take up 107 micrograms of cyanide. The most, if you took the fish and you hollowed the fish out and you made them a plastic bag or a balloon, and then you filled them up with that 25 part per million solution of salt water, right? So you got rid of all the mussels and everything else in the fish, and it was just a container holding that liquid. The most that you could put inside that container would be 25 micrograms of cyanide, right? So that obviously caused a lot of concern, because the 107 is the absolute minimum amount that could have been in those fish to get that graph, right? That's not even in the realm of possibility. So then I calculated a dose. So when you go to the doctor and get medication, or you take aspirin or Tylenol or Advil, right? There's a dose calculation when they tell you how much you can take a day, and you do not want to exceed the recommended dose. Obviously, there is no recommended dose for cyanide because it's a poison. But we know from the weight of the fish and some calculations that the dose that these fish would have gotten are orders of magnitude higher than what pretty much any animal we know of, right, can survive an acute exposure of, right? If you give an animal an acute exposure, so they either ingest a large amount or they are immersed in a bath with a high dose that was this high, right? We don't know of any animal that would survive that. I'm sure there's an edge case here where an animal is consuming something high in cyanide and they might have some extra defense mechanisms. But if you look in the literature, pretty much 4 milligrams per kilogram kills everything. And we're orders of magnitude. So we're at 78.5 milligrams per kilogram on the dose. So we knew it was impossible. Right? Now, you could not believe our math, right? You could say, okay, I just don't like math. I'm not gonna pay attention to the math here. But an important point here is that if you look back at the graph, and I'm going to walk you through this data, you'll notice a few things are missing. This data is really beautiful, right? So you'll see this little fuzzy tick up here on that red box. Kevin, can you see my arrow, the laser pointer shows? Okay, there's no way I can see it. So if you see this little fuzz right here at the top of this arrow bar, you'll notice that that is called an error bar, that little fuzz right there, right? And you'll notice over here that pretty much an absence of one there. And that gives you an idea of how much difference there is between every fish that they tested. So humans are all different. We respond to things differently, right? You wouldn't expect us all to respond to the same thing the same exact way. Even my twins, they don't respond to the same thing the same way. You expect some variation. And there is no variation in this graph. And I sat down, my undergrads down when we were getting ready to work on this project, and I said, look at this beautiful data here. This data is so beautiful. This study is going to be so easy to reproduce. Just to give you an idea of what you would expect to see an error bar look like in some type of data like this, is you would expect to see a large amount of variation, right? Because the fish, right, are being exposed to a toxin, right? And they're going to take up that toxin at different levels. And you would expect to see a pretty high level of variation. No idea what that would be in this case, but you would expect it to be pretty high. Definitely not near zero. Right? Even more disturbing is the time between these samples is a week. So 14 days on there, 21 days, and 28 days. And you'll notice that in the low treatment and also in the high treatment, that the amount of thiocyanate that was reported leaving the fish remained almost identical over a 7-day span, right? So that's really hard to get your mind around. And as troubling as that is that if you look at the increase that they observed at 14 days to 21 days, you will notice that if you take this line and you just move it down to this graph, which is all I did, right? The slopes of those lines are basically identical, right? And that means that the low treatment and the high treatment saw the exact same rate of increase, right? Between those 7-day time periods, 14 days after exposure, right? And so this is all really troubling to see in data. And we did not look at it this way until we did the math. So, all right, now that we've jumped out of the rabbit hole that we been down for 4 years trying to replicate somebody's work that was not possible, right? We were really curious what was happening in the fish. And we've published several papers on this. I've given a couple talks. Alex Bonanno gave a poster last year at MACNA. And essentially what happens is the fish are exposed to cyanide, right? And you can see the gray here is the cyanide being converted to thiocyanate on its way up. And then as it's headed down, they get rid of the thiocyanate out of their blood very quickly, right? Very, very quickly. And you can see we actually have a good bit of variation with these error bars. This is very normal for fish. Again, fish all experience this a little differently. They're going to respond to it differently, right? We got some criticism when we did this first study because we didn't use the exact same species that they reported in their 2012 paper. So we went back and we used the same species that they reported. And this manuscript is in review right now. It's finishing up the revision process. And so this is an earlier slide that I had made of that. But you can see, again, these are individual data points from the individual fish. And the Clarkie clowns get rid of the thiocyanate from their blood plasma even faster. So, so it is really not possible for fish to excrete thiocyanate in any measurable quantity to be able to detect it. We believe that the fish probably store the thiocyanate in their muscle tissue. It's called a two-compartment model. We think they take the cyanide in, they convert it to thiocyanate, it's in their blood plasma, it leaves their blood plasma and goes into their muscles, and then it will slowly leak out of the muscles over time, probably. But there is no possible way you can measure this in a tank of water. So I get a lot of questions about this. Did they make it up? What happened? Do you trust science, right? And so I'm gonna spend a couple of minutes here talking a little bit about the process and how science is self-correcting, right? We asked National Geographic to correct the record, and when our paper came out and they published another story, another article about this topic, and I would encourage you to go read that. It gets into the details of some of this quite a bit. But science is a self-correcting, self-policing discipline. Scientists like me that are working on something, when we find something that's kind of unusual, we tend to pick at it pretty hard to try to figure out what's going on, to try to find truth. And that's what we're after. We really want to know what happened. And the best example I can give you that today is this topic of Dr. Jonathan Pruitt's data around social spiders. In January, it became clear to some of his collaborators that his data was not real. And so a senior author on one of the papers discovered this when someone had sent her a request for some raw data, and she went to look at it, and something really looked wrong. And so Dr. Lakowski, she did the honorable thing, and she's an assistant professor at UC Davis. This is a really devastating thing to have to happen to somebody in early career science. And she retracted their paper, right? Because the data was not explainable, right? Which is the correct process in science when a paper has either been published that has fraudulent data or that it's irreproducible or someone cannot explain their data, right? Is to retract the story so people understand that it was not a valid study or there's something really wrong with it. So is cyanide detection possible? Is it a myth or reality? You know, we went down quite a rabbit hole. And then when we came out of the rabbit hole, we learned a lot about what's happening in the fish. And now we're going back to the future, right? So in 2009, I had started working on gene regulation and what was actually happening genetically. So when you expose these fish to these toxins, their body has to deal with it. There's a lot of enzymes and things that are made. Those are made by genes, right? We looked at the gene regulation. A colleague of mine, a geneticist here at Roger Williams, Dr. Carrie Warren, and I were working on this pretty hard, and we shelved this and put this on the shelf because this beautiful study came out in 2012 that made it look so easy just to look at the water. But we've picked this back up with Mystic Aquarium, and we've got some really exciting preliminary data. And so from there, we're hoping to be able to generate some type of assay that would let us look at genes or proteins. We're very early into this, but we have a manuscript that's being produced now on this early data, and we have some more funding to continue on with this. Very excited about this work. So yes, cyanide fishing is still a problem, in case you're wondering. I can't tell you how big of a problem it is, but it's a big enough problem where you should worry about it. It's a real problem in the food fish side of things, where they collect live food fish for live seafood markets in Southeast Asia, but there's still aquarium fish collected with cyanide. The 2012 paper that I mentioned, Vaz et al., has irreproducible data in it. We weren't there. I wasn't there when the chemist, and neither was the senior author or the lead author of the paper. They never saw this chemistry being run either. We weren't there when the chemists ran this through the instrument, so we can't tell you what they did, but I can tell you it's not reproducible. So it's either highly flawed or it's fraudulent. Right? Umberger and Downs presented data based on the same method, right? And we know that that's irreproducible. And they presented data, and I've got a little snapshot here of their data for you. Their numbers are even more unbelievable than the numbers that Collato's group reported. They're an order of magnitude higher right, than Collato's group. And so we know that the method doesn't work. And then we also know these activists have an agenda when it comes to why they're published in these white papers. And you'll notice they've never published a peer-reviewed paper with this. They just put a press release out. And their data is really suspect. So I think we can discount all of that as there's some serious flaws and challenges there. And certainly this data is not real, right? Fish were not exposed to cyanide and generated that data. That is not possible. Something happened. So many of you are wondering what you could do. What can you do knowing this, having this knowledge? How are you better consumers? How do you not buy fish that are caught with cyanide? I, for many years, have been buying fish from vendors that I'm very comfortable with. I buy fish from Live Aquaria. We get fish from Quality Marine. We get fish from some other vendors that we're really, really comfortable with because they know their supply chain. And that's my biggest suggestion is know your supply chain, buy fish you can from short supply chains. And that's what you can do right now. Where we're trying to go with this in the future is we're trying to make some semblance of an assay or a test where we can give people a certification on their supply chain or possibly a non-lethal test so people could have their fish tested. We still have quite a bit of work to do on this. This stuff is not easy, but it's not impossible. And we need more scientists working on this topic. I think when people think problems are solved, they stop working on them. And that happened a little bit here. We got to the point where we were so frustrated that we decided we were going to continue and figure out what actually happened. And once we could figure that out, then we would be able to continue forward. And that's where we're at today. So with that, I want to thank you very much. I want to thank Kevin Erickson and MACNA for hosting this. This has been a really nice MACNA. I know it's been really disruptive, but MACNA does some wonderful things. The student scholarship they do and the community engagement they work on is really important for supporting a sustainable industry. And events like this are really really valuable to the aquarium industry, but also to scientists who are very interested in studying organisms that they need to keep in captivity. And I'd be happy to answer any questions in the panel discussion. Thank you, Kevin.
Thank you very much, Andy. And if you have questions for Andy, you can go to macna.org/questions and we will get those, and he'll be at the Q&A later today. Now, Andy, I have a quick question. Oftentimes we receive emails or messages at MACNA, and they're concerned that they might have a fish collected by cyanide or with cyanide methods. So what's the kind of likelihood of that? And, is that prevalent in the United States now?
Well, it depends on where their fish came from, right? So if they're getting fish from Fiji or from the small South Pacific island countries or the Caribbean, it's just not something that they have to worry about. Their fish come from Indonesia or the Philippines. There is some level of cyanide fishing there. And so there's reason to have a little bit of concern. We don't know the exact percentage of fish coming in the country. It's probably lower now than it had been at the peak in the 70s, 80s, and 90s. But we don't think it's at a level where it's unlikely that somebody would buy fish that were cyanide free from those countries. It is likely, if you buy enough fish from enough vendors, that you would find some fish that were caught with cyanide. And I think most people think this because their fish died, right? And they're trying to figure out why did their fish die. There's lots of things that go into it. I can tell you that we expose a lot of fish to cyanide in our lab in the past. These are done under very strict and very controlled conditions. And we try not to kill any fish. But if you give them too much, and I mean just over the amount we're using, then you end up having some mortalities. And so it's definitely a concern. And I think hobbyist education is really important here. And working with the supply chain to try to reduce that is also extremely important.
Andy, in theory, should a hobbyist be able to go to a store and say, how long is your supply chain? How many steps between the collection and to the individual hobbyist?
Well, that's exactly what they should be doing, right? Those questions are really informative to someone. So if you walk into a fish store and you ask those questions, you should expect really good answers, right? You should not expect, well, we have a great supply chain. Our vendors are the best. There's never any problems. You should expect them to explain to you how the fish got to them and where they came from, because the importer or the distributor they're buying from, if they're a reputable distributor, right, they will have that information. Some of the distributors have information, right, where you can actually scan a QR code and actually see when that fish landed into Los Angeles, right, and where the fish came from. That is a really powerful tool, and those types of tools really need to be encouraged.
Thank you once again, Andy. I look forward to the continuation of your research and having you out at future MACNAs.
Thank you so much, Kevin.